and welcome to my presentation on recombinant DNA techniques in medicine. So I'm first going to talk about Sanger sequencing. So this is just regular DNA synthesis, 5' to 3'. So looking at a nucleotide such as adenosine, you can see that on the second carbon, there's no OH group, and that's just what makes the DNA different from RNA, because RNA usually has an OH group there. In Sanger sequencing, not only is there an absence of the OH group on the 2' carbon, there is an absence of the OH group on the 3' carbon. So it goes from deoxy-ATP to dideoxy-ATP. And that's just going to prevent DNA synthesis to continue because there's no 3' carbon to keep adding nucleotides to. So we're going to have 4 different vessels with each different dideoxynucleotide, and we're just going to look and see what's happening in the one with DDATP. So for our ingredients, we of course have the DDATP, but we also have the regular ATPs and the regular guanine, cytosines, and thymines. And we're just going to add or synthesize the DNA until it reaches the first DDATP, and then it will fall off. And then synthesis is going to continue happening again. And this time, instead of the DDATP being added there, maybe a regular A is added, and then synthesis continues until the next DDATP inserts itself into the sequence. So here it is at the next A, and then it falls off again, and then you do it again, and maybe the regular ATPs that are there will be inserted, and then further along a DDATP is added, and then it falls off, etc. So you're going to have different lengths depending on where the A's are. So we're going to do gel electrophoresis to separate the DNA fragments based off size. And you run the gel from negative to positive, and DNA will naturally go towards the positive because it has the negative phosphate groups. And first you're going to add the ladder. That's just fragments of known sizes that you can compare to. And then in the other wells, the first one, you're going to put the DDATP reaction solution. So in each of those wells, you'll just see that, oh, it ends in A, or this fragment ends in G, C, or T. And then you're going to start running your gel. The shorter DNA fragments are going to sift through the gel faster than the larger ones, so you'll see them separate out like that. So as you can see, the bottommost band is in the G column. So the first nucleotide in the sequence is G, and it's going to be on the 5' end, and that's because DNA synthesis occurs 5' to 3', so it's G. Then the second from the bottom is going to be the T band, as you see there, and you just put T next, and then it's going to be G again, and then G again, and you go and you see, oh, A is next. So we are going to take our sequence, and we find the complementary bases to find the original template strand. So our strand that we synthesize is complementary to the strand that we were reading from in the beginning. And what do you know? The exon that we are looking at is for sickle cell anemia. Specifically, this exon is part of the beta globin gene, and normally it is supposed to be an A in the wild type, which will code for glutamine, but in sickle cell anemia, it is a T instead, and so that is going to code for a valine instead. So the second DNA sequencing technique that we're going to go over is the high throughput sequencing technique, or also known in our biochemistry book as next generation sequencing. Hence the Star Trek music, next generation, whoop whoop. And what makes this technique special is that it is a lot faster than Sanger sequencing. And I like to think of it as, oh, the spaceship's going warp speed, so it's a lot faster. And the reason for this is that we're able to sequence a bunch of DNA fragments at the same time. So we're going to take our genome and we're going to put it in the blender, chop it all up. Now that you've chopped up your genome, we're going to pour it on the side along with primers of known sequences, also known as adapters, which are the purple things, and that helps stick the DNA fragments to the slide as well. And then you replicate those fragments, and then you sequence all of them at the same time. And then sequencing is happening one nucleotide at a time, like Sanger sequencing, but the difference is that it's happening at all the fragments at the same time. So it's a lot quicker than just doing one. So after sequencing all the different fragments of DNA, you put them in order by matching up the overlapping ends and using a reference genome to match against as well. So I'm going to move on to fluorescence in situ hybridization. We are going to take chromosomes from interphase
metaphase and prophase, and we're going to label them using a fluorescent probe that is complementary to the specific sequence that we are looking for. You can use many different colors, and you can label a whole specific chromosome, or you can have a probe that is specific just for one type of gene, such as the blue one here.
So for chromosomes, you should only see two of each in each cell. So you should only see two red dots. And for the genes, usually you should only see two as well for the homologous pairs. This is very useful in detecting extra or deleted copies of a gene or chromosome. Let's say that we label a different chromosome yellow and we want to see if there's any translocation. You can see something like this happen too. And you can also use FISH for spectral karyotyping, which is having a specific color for all the different chromosomes. And you can see the rearrangement or translocation that I was talking about before between chromosome 2 and 22. Thanks for watching my presentation. I'm sorry if it was a little rushed. Tried to fit in as much as I could.